welcome to all of you who've joined. I'm Lisa Crump. I'm a veterinary epidemiologist at Swiss Tropical and Public Health Institute in Allschwil, Switzerland. And I'm here with my colleague, Jakob Zinstad, there you go. I'll turn it over to you, Jakob, to welcome.
Hello, everybody. I'm Jakob Zinstag. I'm a veterinary epidemiologist, and I'm also, together with Lisa, we are editors-in-chief of the CABI One Health platform, which includes also the One Health Knowledge Bank. And the Knowledge Bank actually should assemble all what we know about One Health. And I'm very grateful to the organizers, to our colleagues from CABI, that they organized this first webinar on the importance of antimicrobial resistance from a One Health perspective. Please, Lisa.
So today we're going to, next slide, please. Today we have a series of 3 different sessions, and we have speakers in each session, as you've seen on the agenda. And so we'll allow each speaker to present their material, and then we'll move to a question and answer section after each session. And you can feel free to go ahead and type your questions into the Q&A boxes, which you'll see at the bottom of your screen. If you want to go ahead and chat with each other and introduce yourself and network, you can do that in the webinar chat. Please just use the Q&A for the questions that you have, and we'll be following those questions. If you want to direct your question to a particular speaker, you can type the speaker's name in. Otherwise, we'll open it to the floor of speakers. And just for protocol, the Q&A is only for questions, and the webinar chat is for chatting with each other. So I think that should be clear. I see that we have a couple of questions already in the chat, and we'll be looking for you to go ahead with that. We'll start with next slide, please. I think we advanced a little bit too far. Jakob was going to present just one slide.
I hope you have it in the deck, or should we share it?
I don't think you can share it.
No, I cannot share it. Otherwise, I can also just explain it.
Sure, go ahead.
So, formally, welcome to that webinar on antimicrobial resistance. And here it is. Thank you very much. Exactly, this is what we wanted. Antimicrobial resistance are actually a very complex problem, and we are very grateful to all the speakers today who show us the different facets of this. And from a One Health perspective, we try to bring this all together. And I would say one of the most advanced institutions of surveillance of antimicrobial resistance is actually the Canadian Program for Antimicrobial Resistance Surveillance. The so-called CPARS actually is one authority that does the antimicrobial resistance surveillance in human patients. You see that on the bottom right, but also in the whole food chain, in the food animals, but also in the whole environment, in wildlife, sewage, soils, surface waters, and basically the whole, what we would call, social ecological system. So with this metaphor, we don't want to unify a dogmatic approach to it, but we would like to ask all the people that speak today to tell us what they think from their perspective would be the advantage of working closer together between human and animal health, environmental sciences, agriculture, and all other domains that are involved in questions of antimicrobial resistance. We don't want to be longer, and we invite quickly the first speaker. Please change slides.
Next slide, please.
Next slide, please. Exactly. Professor Craig McLean, we are very pleased to have you here today. And I would say what you will speak about, the evolutionary dimension of AMR, is absolutely a crucial understanding of the underlying processes. Please welcome to this webinar, and please, the floor is yours.
Thank you very much, Jakob and Lisa. I'm trying to share my screen
McLean,
now.
are you online?
Okay. Good. Can everybody hear me?
Yes.
Yes? Okay, good. Thank you.
Don't hear you.
So good. Thank you. So my name is Craig McLean, and what we study in my research group is the evolutionary biology of antibiotic resistance. And so today I'm going to talk to you about the work
we don't hear you. Sorry.
You don't hear me?
No, we can hear.
You cannot hear. Okay, hold on one second.
I can hear well.
I can as well.
I can hear and see.
Yep.
Okay.
It's working very well.
Greg, is your microphone unmuted?
Yes, my microphone is unmuted.
We can all hear him.
Yeah, I think other people can hear me. Jacob or Jakob, could it be that you're... Can you hear me now?
Yes, I can hear you very well.
Okay. I'm not really sure what the problem is, because obviously Lisa and Jakob can't hear me. Lisa, Jakob, if you can hear me, can you show me a thumbs up?
Yes.
We have you now.
Okay, good. Okay. Right. So technical glitches aside, on to AMR. Okay, so I wasn't really sure where to start in my introduction, so I thought I would start here and start off by saying that really we know now that probably about half the humans who've ever lived have died because of infectious disease. And throughout human history, bacterial infections have been a leading cause of disease and death in human societies. So this painting, for example, which I like to show, was painted by Bruegel the Elder during one of the great waves of plague that swept through Europe in the Renaissance. And this plague killed about half the people living in Antwerp, where Bruegel was based. And the interesting thing is it shows not only the disease and death caused by plague, but also the social upheaval, which I think is actually one interesting aspect of disease that is sometimes overlooked. Of course, we know now that we're in the danger, there's a real danger of returning to the dystopian world depicted by Bruegel. We're in a situation where AMR infections now cause somewhere between 1 and 5 million deaths per year, and that there's a kind of realistic possibility that by the mid-21st century, AMR infections are going to cause something like 10 million deaths per year. I think that AMR really is one of the biggest challenges facing humanity in the 21st century. And my background really is as an evolutionary biologist. Okay, I trained really in very blue skies evolutionary biology, and I became interested in AMR about 12 years ago. And at the time, this was a very simple cartoon that I sketched that views how evolutionary biologists think about AMR. And the idea here is that we've got some sensitive population, and we expose it to antibiotics, and that kills off most of the bacteria. But there's some rare resistant variants, and they can continue to survive and grow. Okay, so we see that the sensitive population are replaced by resistant bacteria. And this is really just a really simple and elegant example of Darwinian evolution by natural selection. Okay, and so then when kind of the next step that evolutionary approaches to AMR start to think about is how resistance can be stably maintained in pathogen populations. So I've deliberately drawn the resistant bacteria here as being small, and that's because what we find is that resistance usually is accompanied by fitness costs. So when bacteria become resistant to an antibiotic, they pay a price for this. And we can see this in terms of reduced growth rate, reduced virulence, etc. And because of this, it's hard to understand, that bacteria live in a world where kind of antibiotic exposure is quite discontinuous, and it's hard to understand how resistance can be maintained. And the idea is now, for example, if bacteria encounter an antibiotic-free environment, for example, like if you have a pathogen that's transmitting between hosts, that these low fitness resistant bacteria can simply be outcompeted by sensitive competitors. And so one of the big challenges in evolutionary model and so this is important, because what this tells us is that actually if we stop using antibiotics, resistance should go away. But I think one of the really key insights from evolutionary biology has been that sometimes what we observe is that actually these bacteria, resistant bacteria, evolve compensatory adaptations. Okay, and these let them be antibiotic resistant, but not pay any fitness penalty for it. And when this happens, we can have resistance being stably maintained in pathogen populations, even without any antibiotic use. So this cartoon that I'm showing you basically comes from, theoretical mathematical models and from lab experiments. And there isn't really very much evidence of this, certainly not understanding what stabilizes resistance in real-world pathogen populations. And what my lab has tried to do over the last 5 to 7 years is try and bridge this gap by asking questions like what stabilizes resistance, not in simplified experimental systems that we've worked on for years, but in the real world. And we work on this problem across a variety of pathogens. We're much more interested in the questions than in the bugs. But today I'll be talking about some work that we've done with E. coli. So we're going to jump now to colistin. So colistin was one of the first antibiotics to be discovered. And when it was discovered, it was realized that it was quite a potent antibiotic against gram-negative bacteria. Sorry, there's some feedback. Can I ask people to turn off their microphones? Thank you. So quite potent antibiotic against gram-negative bacteria, but with some really undesirable side effects like nephrotoxicity. And so it effectively became, but it was cheap to make, and it effectively became a veterinary antibiotic. It's something that was used at a very large scale in agriculture, okay, particularly because including it as a growth promoter in animal food is kind of economically beneficial for farmers. But what's happened, so it was used started to be used at a very large scale. But what's happened is that over time, as resistance to other antibiotics has spread, colistin has become an important last line of defense for treating infections caused by multi-drug resistant bacteria. And so we're left in this kind of absurd situation where we have an antibiotic that's being used at a very large scale in agriculture, for example, as a growth promoter, is also a last resort antibiotic for serious infections. And amazingly, okay, so colistin was actually used as a growth promoter for decades without really very much appreciable resistance. And so in China in the kind of 2010s, all of a sudden resistance to colistin in E. coli really shot up. And this was mediated by a gene called MCR, mobile colistin resistance 1. Okay. And the Chinese government reacted to this by banning the use of colistin as a growth promoter. So this was a kind of very interesting ban, and it reduced the consumption of colistin in agriculture by about 90%. And so this gives us a really amazing system to test, about what can stably maintain resistance. And I want to come back actually now to this point that Jakob made in his introduction, where he showed this very complicated map with all of these kind of connected One Health reservoirs of AMR. And when MCR1 was discovered, okay, even though colistin was mainly being used in agriculture, MCR1 was found basically in all of these different reservoirs. And one of the really interesting things is that actually when the use of colistin was banned in agriculture as a growth promoter, so there was a massive reduction in agricultural consumption, the frequency of colistin resistance declined not only in agriculture, but actually across those sectors, showing that in this case, it really was agricultural use of colistin that was driving the spread of resistance across humans, the environment, all of these different kind of One Health compartments. but I haven't been involved in this part. So what my lab has done, and I really want to highlight the work here done by Lois Ogunlana, Pramod Jangir, Liam Shaw, and Celia Souk, is to try and understand how resistance could be maintained. Okay. And colistin is a peptide, okay, and it's strongly positively charged. And bacteria, like E. coli, are strongly negatively charged. And the mechanisms by which MCR1 protects bacteria are a little bit more nuanced and subtle than this. But one of the important kind of things that MCR1 does is it reduces the cell surface charge. And this then therefore reduces the attraction of colistin to bacteria. And so to understand the fitness costs of this, we took a kind of resistant strain of E. coli and a sensitive strain and competed them against each other just in lab culture media. And this is showing relative fitness. Okay, so this is there's no colistin around, just in the absence of colistin. What is the fitness of bacteria with MCR1 as we increase MCR1 expression? And what you can see from this is that MCR1 is very costly. Okay, so high levels of expression, it's actually reducing bacterial fitness by about 50%. So to put this in context, a typical resistance gene costs a few percent, okay, for bacteria to express. MCR1 can cost up to 50%. These are some of the biggest costs of resistance that have ever been reported in the literature. So given this, you'd think that if we reduce colistin consumption, MCR1 should effectively disappear very quickly. Okay? But this isn't what's happened. So how can MCR1 persist given its costs? So we started looking to try and test this idea that effectively that the gene has evolved to become more stable. And through surveillance programs, one of the real advantages of surveillance programs is that we have thousands of sequences of MCR1. And one of the things we noticed, there's not very much genetic variation in the gene, but if we look at the region upstream of the gene, so this is the nucleotide sequence just upstream of the gene, and this here, what's showing here, are regions of this promoter that are really crucial for the expression of the gene. And we found that actually there's some, that they're kind of enriched in polymorphisms. So it's common for bacteria harboring MCR1 to have mutations in this regulatory region. And we then cloned these mutations. We then made a little like toy MCR1 expression vector where we've got MCR1, and it can be under the control of the wild type regulatory sequence that MCR1 initially had, or we can clone in one of these mutations that we find in the kind of real world. And so what this shows now is the fitness. Again, this is in colistin-free media. So this is a wild type control strain that has no MCR1. We're going to just arbitrarily say it has a fitness equal to 1. So under these conditions, MCR1, under its at endogenous levels of expression, it reduces fitness by about 30%. And what we can see is these mutants tend to increase fitness, okay? So they're still colistin resistant. Actually, if anything, they tend to be a little bit more colistin resistant than the kind of ancestral MCR1, but they have higher fitness. So these mutations in the regulatory region allow E. coli to be as resistant, if not more resistant to colistin, but to now pay a smaller cost for this. And this is just showing, don't pay too much attention to this, but this is just showing basically that the way this happens is that these mutations actually effectively tune down the expression of MCR1. So these mutations reduce the levels of expression. So this would be the kind of wild type MCR1 regulatory sequence. These mutations reduce the expression level and they increase fitness in the absence of colistin. Okay, and this is quite a strong trend.
Craig, can I just ask you to wrap up quickly? I'm sorry, but we're a bit over time. If you can just get to your
Okay, we're already over time. Okay, that's
Sorry.
fine. Sorry. Okay. So I'm gonna end here quickly. Okay, so these mutations are on plasmids. Okay, so MCR1 is naturally carried around on plasmids. And what we find is actually these plasmids have moved between strains of E. coli. Okay. And so what this has done, so forget this graph, we'll just stick with this picture. The important thing is what this has done is it's disseminated these regulatory mutations that increase fitness across the diversity of E. coli. And if we think now about the impact that this has had on the band, okay, so this is showing the frequency of carriage in farms. This is from kind of One Health surveillance in China of wild type E. coli with a wild type MCR1 or with the most common promoter mutation. We see that what the ban has effectively done is, yes, there's been a reduction in the overall prevalence of MCR1, but really what that ban has done is it's wiped out the E. coli with a wild type regulatory sequence. And some of these mutants that have an altered regulation really have not been very heavily affected by the ban. So what we see is that this evolution has reduced the costs of MCR1, and that has stabilized MCR1, allowing it to persist even after colistin use has been dramatically decreased. Okay, and so what this shows is that how evolution can eliminate the cost of AMR genes. AMR can be stable even at low levels of antibiotic use, and what it really highlights is the need to actively eliminate AMR from pathogen populations. It tells us that sometimes just reducing consumption is not going to be enough. So apologies that I went over time.
Thank you very much. I'm sorry to rush you there, and we really appreciate the presentation. We're going to go ahead and pass over now to Dr. Fergus Allerton of the BSA VA Willows Veterinary Center. Dr. Allerton, over to you.
Thank you very much, and for this opportunity. Can we go to the next slide, please? And the next slide. So I wanted to try and talk about the guidelines that are being used for antibiotic use recommendations in small animals. And I recognize this is very much a One Health topic, and I work in something of a niche field in working with just cats and dogs. But I think lots of the ways that we are going about trying to improve and optimize antibiotic use in veterinarians and in human clinicians, there's a lot of overlap here. And it was interesting hearing about the effects of a formulary restriction on a ban of the use of antibiotics in China. But currently in the UK, we are operating with enablement approaches to stewardship. We are trying to encourage clinicians to make evidence-based selections and decisions to minimize and optimize their antibiotic use. And the Protect Me tool, which is freely available for download, and any veterinarian working in small animal practice, I would encourage you to have a look at this and see if you can introduce some of these recommendations into your daily practice. Next slide, please. Yeah, perfect. And the Protect Me is actually an acronym, and the latest iteration has introduced some slight variations on these themes, but I think that these echo across all spheres of antibiotic use. So we really are encouraging vets to prescribe antibiotics only when necessary, to really ensure that any decision to use antibiotics is based on evidence of bacterial involvement and clinical requirement. So there are lots of bacterial infections that will get better without recourse to antibiotics. So we are trying to encourage selection when that threshold of significant requirement is breached. We want vets to think about using other non-antibiotic treatments. Can they get away with anti-inflammatories and afford the animal time to get better of its own accord? There's a lot of information in both human and veterinary medicine as to how long should we treat for, and we'd love that to be evidence-based. We're doing some studies now to try and establish what the optimal duration of antibiotic use would be for dogs with cystitis, and extrapolating from findings in human medicine, it is likely that shorter courses are non-inferior, and therefore less antibiotic going into the environment. It would seem likely that shorter is better in that context. And I think we're always encouraging vets to try and establish the etiology and demonstrate that there is bacterial involvement and therefore a requirement for using antibiotics. And using tools like cytology and culture can help increase confidence as to that need. We also want vets to audit their antibiotic use and those practices, so that they can reflect when those antibiotics are used necessarily and when that use may be inappropriate, to again look for ways to improve it. And we want to educate, we want to share this message, because where a vet is recommending that antibiotics may not be necessary to treat a dog or a cat, these same principles apply when the owner may find themselves at the doctor having that same conversation as to whether antibiotics are required to treat the owner. And if we can share a similar message, hopefully this will resonate and increase public awareness of the importance of AMR and antimicrobial stewardship in general. And I've just mentioned the antibiotic amnesty here, which is an ongoing initiative where we're encouraging pet owners, in the same way that human doctors and pharmacies are encouraging patients, to return any leftover unused antibiotics back to the vets, back to the pharmacies, to avoid environmental contamination and pollution, because all of these things will contribute potentially to driving more antibiotic resistance. Next slide, please. Next slide. And that is the owner education part, I think, really has these big One Health ramifications. So we have created a little video, I'll show that in a moment, but really what it's trying to do is signpost owners to additional information, improve their awareness of AMR. Vets often feel that owners are the driving force for an antibiotic prescription. We feel under pressure to offer this treatment to our clients, but if we could get owners to be part of the solution, if we could encourage them to accept where antibiotics aren't necessary, that they shouldn't be prescribed, maybe together we can reduce our antibiotic use. So owners, for me, are often an overlooked stakeholder and probably one of the most important opportunities for us to concentrate on and improve our antibiotic use. I won't show the whole of the video, but if you'd like to go to the next slide and press play, we can just dip into that and see how this tool could work. And it does actually have a narration, so there are versions that are available with the voice, but as you can see, all the words are written out as well. We have translated this now into 25 different languages. It does cover a common scenario, something that vets will encounter quite frequently, of a dog going in with acute diarrhea. The dog seems systemically well, but the owners feel that antibiotics are required. And it's up to the vet really to convert this consultation, rather than into an owner leaving with a pot of pills, as they may have done previously, into being better informed and hopefully leaving with other ways to try and manage that case. And we did run this as a randomized controlled trial, and we showed that just watching this 2-minute video could convert or could alter owner opinions and made them less likely to request antibiotics, increase their awareness of AMR. So this tool is freely available. It's on the eNavat website, and I would encourage any vets in practice to download it and maybe incorporate it into their working environment, whether it's showing it in the waiting room or encouraging owners to access it while they're waiting for their consultation. We can move on now to the next slide anyway. And I won't go through all the sections of the new Protect Me box in too much detail, but I will say that one of the biggest parts of the poster is trying to highlight situations where we don't need antibiotics, and gastrointestinal infections is a good example of those. Antibiotics are unlikely to make a significant benefit in dogs and cats with acute vomiting, acute diarrhea, pancreatitis, chronic diarrhea. And if we could reassure vets that actually those animals will get better without recourse to antibiotics, hopefully we can bring down overall use. These are very common presentations, and we know that 50 to 80% of vets will use antibiotics when faced with acute diarrhea. That number should be less than 5%. So these are the opportunities for big wins. These are situations where you could say, instead of reaching for antibiotics, let's find better ways and bring down that antibiotic use. Next slide, please. I think we'll just jump through a couple of these if that's okay. Next slide, next slide. So in essence, the poster does convey a lot of different information about various organ systems. We've got information on surgical prophylaxis, managing wounds, urinary tract, respiratory tract infections, and advice around skin and ear disease. And hopefully, if vets adhered to the Protect Me guidance, we would work together to reduce our antibiotic use. So if there are vets online now, I would really encourage you to go to the BSAVA website and look up the latest Protect Me poster and incorporate it into your practice policy. I've mentioned the antibiotic amnesty, and if there are opportunities, if vets or pharmacies can encourage their owners and their patients to return unused or leftover antibiotic, that will prevent it being used again inappropriately and will also help reduce environmental pollution. And do think about downloading that video if it could be of use to you to help educate owners about AMR. Thank you very much.
Thank you very much, Fergus. I will continue with this one, the use of medicine in agriculture. Please, continue.
Good afternoon, everybody. I didn't quite hear the introduction, but I'm assuming that was a green light for me to crack on. So my name's Chris Lloyd. I work for the Responsible Use of Medicines in Agriculture called RUMA. Next slide, please. Next slide. Thank you, that one. So RUMA is an alliance of 26 organisations across the UK food supply chain, which is dedicated to ensuring and promoting the responsible use of veterinary medicines in UK farmed livestock. We've had a recent focus on antibiotic resistance. In 2020, we set up a sister organisation called RUMA Companion Animals and Equine, and Fergus is an active member of that sister organisation. Next slide, please. I've been asked today to talk about how UK livestock fits into the One Health AMR agenda. And in the UK, it's very much a One Health approach, looking at how we use medicines in humans, farmed animals, companion animals, and the environment. And the Lord O'Neill report has been a real catalyst for a lot of that One Health activity. Next slide, please. A couple of years ago, FAO did a case study on the UK's livestock sector actions against antimicrobial resistance. And this, I would recommend it to anyone who hasn't seen it. It's on the FAO website. And this page just really sort of charts that journey from the early days back in 2010, when Europe collated its first set of antibiotic use data under SFAC. 2011, the British Poultry Council set up the first antibiotic stewardship group, which was the first sector in the UK to do so. Then we fast forward to 2015, and the pig sector set up the next antibiotic stewardship group, realising the challenge that they faced in the pig industry. The Lord O'Neill report to government set out the AMR challenge and challenged both human medicine and the farming industry to make their contribution against this increasing issue for public health. In 2016, RUMA convened the first meeting of the RUMA Targets Task Force, which is a group of a vet and a farmer from every sector, every livestock sector across the UK, who are charged with working with their stakeholders back in their sectors to create a series of targets which that sector would work towards in terms of reducing their antibiotic use. Next slide, please. So 3 years, we're now in the second phase of the RUMA Targets Task Force. And we publish our report in November of each year, and that complements the UK's VMD veterinary antibiotic resistance and sales surveillance report. So the VMD, the VARS, as we call it, is a record of the sales of antibiotics and the abattoir surveillance in the pig and poultry sector for antimicrobial resistant bacteria. And the rumour report complements that by explaining all the industry activity that's going on to deliver our targets, but is contributing to that progress that the VARS report, that the antibiotic sales that the VARS report reports. Next slide, please. So what have we achieved? Since 2014, we've reduced total antibiotic use in the livestock sector, the farmed livestock sector, by 59%. Next slide, please. We've also reduced the use of HPCIAs, which I'm sure all of you are aware of, the antibiotics of importance for human use. We've reduced those by 82%. And just in reference to Craig's presentation earlier on, we no longer use colistin without any ban. That's a voluntary withdrawal of colistin across all the sectors. And I think our use in 2022 was 0. Next slide, please. So in total, we now use less than 25% of the UK's total antibiotic sales in farmed livestock, despite producing over 1 billion farm animals to feed our population. Next slide, please. So this is a slide from the VARS report that kind of runs in parallel with that reduced antibiotic use. We're now seeing an increase in susceptible bacteria. So this is surveillance, harmonized surveillance that is carried out at the point of slaughter in abattoirs in pigs and poultry. The green arrow on the left indicates the increase in susceptibility, and the green arrow on the right indicates the reducing level of drug-resistant bacteria. So that seems to run in parallel with our reduced use of antibiotics. Next slide, please. So as I said, we produced our Targets Task Force report. We're now coming to the end of our second phase, which will complete next year. Next slide, please. Just wanted to give you a sort of a quick summary. So the important thing we learn in engaging with all of the sectors is they're all in different places in their journey of antimicrobial kind of activity. You've got the poultry industry, which, as I said, has been active since 2011, and the pig industry since 2015. But some sectors, when we engaged with them back in 2016, 2017, it was a new issue for them. It wasn't a challenge, but they've all come on board. So in this last report, we've seen reductions in the salmon, pig, and game bird sector. We're now seeing some of those sectors that have been at this for a long time, like the poultry sector. They've now reduced their levels to what would be considered a sustainable use of antibiotics in their sector, remembering that our objective is not zero use of antibiotics, it's responsible and sustainable use of antibiotics, so that we're preventing the buildup of resistance to these products, these medicines, and preserving their use for future generations. In the ruminant sectors, I explained that for some sectors it was a new issue, and in 2017 that was certainly the case in the ruminant sectors. Much more challenging to collect data, much bigger sectors, not so integrated, with a number of small independent businesses. But this year we saw the first set of data for both the dairy, beef and sheep sectors at a national level. So we can now use that data to drive the actions within those sectors to make change. But we also see anomalies, and in the trout sector, the blue fish there, this year their antibiotic use, or in 2022, we saw their antibiotic use go up, and that was in response to a disease outbreak within some of the trout farms, which they had to deal with because it was a welfare issue. But now that's dealt with, we expect their antibiotic use to come down. So whilst we're monitoring and tracking the information, some sectors or antibiotic use can go up at certain times. Next slide, please. So what have we learned from the process? Where we have data, it helps drive decisions, but there's use of data at a national level, so we can defend the industry, we can demonstrate the industry's journey and the commitment to antimicrobial tackling the antimicrobial resistance, but also at a farm level where the vet and the farm use the antibiotic data they've captured, the antibiotic use on that farm, and discuss that in terms of a flock or a herd health plan for that year and the diseases that they're trying to tackle. Absolutely crucial that you have your vets and your farmers and representative organisations all involved. Everyone's got to have the skin in the game, as they say. If not, you don't get buy-in from across the board. The sectors have to take ownership of their targets because this drives motivations to deliver them. In recent years, vaccination has been a really important part of preventing disease, but in recent years, we've seen a disruption to vaccine supply in the UK, which is worrying looking forward. Environmental issues also show us that as we see a change in climate change, some of the diseases that we're experiencing, some of the impacts on management of livestock and fish can cause changes which make us vulnerable going forward. And just to repeat the point, all sectors are in a different place. They have different structures, different organisations, and they're all in a different point within that AMR journey. But we have a complete commitment across all the sectors in the UK to continue to tackle that challenge. Next slide, please. So just wanted to thank you for the opportunity of letting me share the UK story, and just to repeat that that's under a One Health commitment where we're doing our bit to combat AMR within the UK. Thank you.
Thanks very much to Craig and Fergus and Chris for those interventions. I don't have any, I do have one question in the chat. Nope, it's a thank you. So the link to the video that Fergus showed is in the chat. In the interest of time, I think we'll move right ahead to the next section since we don't have open questions on this one. Thank you very much to the speakers for sharing those details. Next, we have Dr. Andrew Singer from the UK Centre for Ecology and Hydrology, who's going to talk to us a little bit about the environmental dimension of AMR. Andrew, the floor is yours.
Thanks very much. Next slide, please. Okay. Yeah, so this is going to be an extremely fast overview of the environmental dimension of AMR, which could be a 10 times longer talk, but hopefully it won't be. Next slide. So antimicrobial resistance is caused by the use of chemicals used to treat and kill and inhibit the growth of bacteria, which is antibacterials, the antivirals, killed viruses, and then the fungals and the protozoals and the anti-helminths, which kill worms. And the action of all of these are effective until the evolutionary response from using them causes resistance. Now, basically, every chemical could potentially be inhibitory towards the growth of an organism. And there's a short list of chemicals that have been shown to also drive antimicrobial resistance because of their toxic effects. And those are basically most of the metals, lots of biocides, which you might not be surprised about because that's why they exist, pharmaceuticals, herbicides, and pesticides. The list is growing for all of those in the literature. Next slide. Unfortunately, I don't have time to get into why, but that's the extended remix version of this talk. The usage of all these chemicals is basically throughout all of society. Most of the chemicals that are used, they don't decompose in the body if that's where they're being used. Very much. So it can be as much as 90% of the drug that you take is going through your body and comes out the other end in wastewater in this case. And most sources either lead to a sink of being the rivers, your coasts, or the soil, which could be cropland. Next slide. Okay, this is quite a heavy-duty slide, which I'm going to just gloss over it, but it's here to just remind you, or at least inform you, that environmental AMR is actually a fairly new discipline within AMR, and we had to sort of create our own jargon, I guess, to understand AMR in a way that was relevant to selection. In the clinic, they're mostly interested in killing bugs. So they have a minimum inhibitory concentration, and you try to achieve that or higher in your bloodstream. And barring lack of resistance, you'll have an effective treatment. In the environment, we're interested in ensuring that the selection for resistance is not achieved. So what is the concentration, the lowest concentration that will be effectively safe and won't drive resistance, above which that concentration becomes selective and you will start driving resistance. That is a value which is derived from UCAST. So this is a database. And so in number one is the UCAST database, and we sort of plot that across all the species. We then decide how many bacteria we're prepared to save and kill. And then you then put a safety factor on that because you're aware of the fact that the database is probably not 100% representative of every microbe that exists on Earth. And so you're left with environmental concentrations of antibiotics that are theoretically that threshold above which you start selecting for resistance. And for anyone who has a sense of what makes a concentration of antibiotics potent, these are very, very low concentrations. We're talking below a microgram per liter for a lot of antibiotics. Next slide. Right, so then once we have effectively a modeled and an empirical understanding of the relevance of these thresholds, these PNECs, predicted no effect concentrations, we can then start to look at the environment and say, well, are we exceeding these concentrations in the many different environments that we're monitoring? And so here's one study that surveyed the literature and tried to examine the relevance of environmental concentrations. So there's a list of antibiotics in municipal wastewater, hospital wastewater, industrial wastewater, surface water, and drinking water. And at the bottom of each of those, you find the percentage of the samples that were collected in each one of those categories that exceeded these PNECs, these predicted no effect concentrations. So fortunately, drinking water in every case did not exceed it, which you would hope. It was probably all treated drinking water, not sort of borehole water. Surface water is nearly 3% of the river waters exceeded it, and obviously it will change depending on the antibiotic because antibiotics will vary in their usage, they will vary in their environmental persistence, and those pretty much dictate whether you'll find them or not. And so then you can see hospital wastewater and industrial wastewater are the big ones, as far as they're almost always going to exceed, or at least half the time they're going to exceed these effect concentrations, and municipal wastewater, depending on the antibiotic, somewhere around 6% will exceed selection concentrations. That's probably an underestimate, but next slide, please. Right, so here is antibiotic production sites. They used to be all around the world, but now they're mostly in China and India. And concentrations of antibiotics downstream of the production facilities have been measured at levels that are consistent with therapeutic antibiotic use. So like 31 milligrams per liter. Remember from previous slide, I was talking about sub-microgram per liter concentrations being selective. So we're talking solidly 1,000 to 10,000 times higher than selection concentrations. And so there's a growing list of this in the world. Next slide. Just very quickly, want to give you a sense of what else is going on in this space. So we had some Ruma given a talk about the antibiotic usage across the animal sector, fish being just one component, but it's a important component because it represents an area where antibiotics are directly added to the environment as opposed to into the feed. In panel B, there's basically a demonstration of the fact that in the crops that we produce, we use antifungals, and they're pretty much used globally, the same antifungals. And unfortunately, the antifungals that we've been using, the triazoles in particular, drive resistance in a human pathogen called Aspergillus. And it's been demonstrated that 40% of the resistant Aspergillus in this case, the UK, the study was done, had acquired their resistance from the environment. And we're trying, there's a study right now, we're looking at what would be the drivers of that. It might be composting facilities that receive contaminated waste. And there may be some high-use industries. In the Netherlands, the bulb industry is one, an enormous antifungal user. And panel D, for anyone who's keeping track, there's no panel C. There is well, all humans eventually will use an antibiotic at one point, if not for quite a number of points in their life. And in countries where there is more prescribing or more use, whichever the case may be, you tend to find more resistance. And this is a study basically looking at the wastewater from 101 locations, countries, and showing the resistance patterns. And they largely mirror each other because humans are humans, but their usage patterns vary based on availability of the drugs. Next slide, please. Okay, last slide. This is basically just an overall summary of the factors that contribute to AMR in the environment. And so there are global factors which apply to everyone, which is the fact that populations are increasing, which means that more people will get sick and need antibiotics. International travel is much more than it ever has been. It's a subtle point, but basically we don't have any replacement antibiotics. So by losing antibiotic usage through resistance, it makes the problem worse. And the warming planet, there are some suggestions that by having a warming planet, you actually have more AMR. Very briefly, behavioral changes. So basically we use antibiotics in humans, animals, and crops. And that usage is not, well, it's becoming more sustainable, but it's not at the point of being sustainable. Because really, we're just irresponsible. The third one is inequality. So we might sort our problems out in the UK, but there's a whole world out there. And they have investment problems, which is the fourth category, which means that there may not be any sanitation or very poor sanitation and hygiene infrastructure. It means they also have much higher exposure to polluted environments. Both of these will guarantee increased infections, which means the need for antibiotics. And then, of course, clean drinking water is an obvious factor in exposure and then eventually the need for antibiotics. And lastly, as I mentioned, the antibiotic pharmaceutical manufacturing is a big point source, but it's obviously there are geographically quite finite in the sense that there aren't thousands of them around the world. Wastewater is, in fact, one of those huge areas where if we sort that problem out, we probably sort a huge element of environmental AMR. And with that, I probably should stop. So thank you very much.
Andrew, for the quick run through the environmental dimensions, which is, as you correctly point out, is extremely complicated. There is one question directed at you in the Q&A, but I'm going to go ahead and move on with the next presentation, and we'll circle back to that at the end. Our next presentation is a video recorded message. Unfortunately, Philip Taylor wasn't able to be with us today, but he's from the Global Plant Clinic at CABI. He's the coordinator, and so we're going to go ahead with this video message from Philip Taylor.
This opportunity to share some of my work. Hello, my name's Phil Taylor, and I'd like to thank the organisers for giving me this opportunity to share some of my work, and also the broader aspect of why crop production is important in AMR. Well, we're all familiar with pictures such as this, the idea of hospitals and urban use of medicines and sewage sludge contributing to the contamination of our environment with antibiotics. But plants are now included as well.
and I'm just going to be talking about the role of crop production in the whole of the AMR. So I'm very pleased to say that in this recent document, the One Health Priority Research Agenda for Antimicrobial Resistance, as you can see, figure 2 has got plant health very firmly alongside human health and animal health, all within the whole context of the environment. So this change is relatively new. AMR had previously concentrated on antibiotics, particularly in a medical setting, but now it's a much wider remit. And it's very fortunate that lots of different previously siloed disciplines are coming together talking about AMR in a One Health setting. And of course, not only are the bacteria medically important bacteria, there are bacteria in lots of different areas of the world, and also fungi too, all of which are within the remit of AMR, as I've said. So 2 photographs here, one photograph of the Dutch bulb industry and the other one of a dead twig. So what relevance do these pictures have about AMR? Well, looking at the Dutch bulb industry in the first instance, the Dutch bulb industry, as you're probably aware, is a massive industry, and they look after their plants very well, and they don't want them to get diseased. So they put on a lot of chemicals, a lot of fungicides onto the plants to look after them when they're alive. But of course, there's a lot of debris and a lot of old leaves and a lot of waste. And what happens is that when they finished harvesting the bulbs, they compost the waste. And the waste is composted so it can be used as compost for the next year. But of course, the composting is taking place in a fungicide-rich environment because of all the fungicides which were applied to the crop. And unsurprisingly, the farmers were selecting for resistance in the composting microorganisms to the fungicides. And so the fungicides which are used are the FRAC group 3, the DMI fungicides, and here they are, some of them on the right-hand side, the triazole fungicides. And so the fungi in the compost heap were becoming resistant to these chemicals. This wouldn't matter except the fact that Aspergillus fumigatus is a fungus which is commonly found in compost heaps, but it also can cause infections of the lung. So you can see it in 3 different guises here. And so the result of this is that Aspergillus fumigatus has become resistant to the fungicides, and many people are now contracting chest infections which can't properly be treated. And the reason that they can't properly be treated is because the resistance to the agricultural fungicides has crossed over to resistance to the medically important fungicides as well. And you can see the azole names here on the left-hand side in the fungal antifungals used to treat chest infections, and they are the same or very similar chemicals to those on the right, which are used in agriculture. So this is a major problem where there's been crossover resistance from agriculture into medicine. But thinking about more widely, we should think about the whole of AMR just against plant pathogens and the resistance of plant pathogens to fungicides which we use to control them. And we give this sheet out as part of the plant-wise training, and all of the active ingredients are here listed in the main body of the table, and the colors of the fonts indicate which groups they're in. So we encourage farmers to swap between one color to another, or even better, to blend the different colored compounds here to prevent resistance from occurring, which is all part of our attempts to reduce AMR. The other picture of these two is this dead twig on the right-hand side. So what's that got to do with AMR? Well, this is a disease called fire blight, and it's caused by a bacterium called Erwinia amylovera. And although it's banned in the EU, some countries, including the USA, use antibiotics to control this disease. It's a big problem for apple and pear growers. And as you can see, the twig dies and ultimately the tree will die. And so we have been able to get hold of a large data set owned by CABI, and we went through the data set looking for where farming advisors had advised farmers to use antibiotics against their crops. And this was a huge data set from many countries across the world. So we were able to analyze where antibiotics were being used, which antibiotics were being used, and against what, and on which crops. And this was published in a 2020 paper, as you can see here. So this is the record that we have. You can see in some cases we had nearly 180,000 records to look at. And within them, we found a small but substantial amount of antibiotics within each of these data sets. And in some cases in Southeast Asia, we had 1,000 records, all of which contained an antibiotic. And if we look at the globe, we can see which kinds of antibiotics are being used against plant diseases. So on the left-hand side, you can see that the pie chart is mostly green, and that's because in South America and in the Caribbean, they're using casugamycin. In the center of the image, the yellow, green, and red pie, this is Southeast Asia, and in the antibiotics of choice here are casugamycin, which is the green, but also tetracycline and streptomycin as well. And then on the right-hand side, we have the Pacific region, and they're using, again, casugamycin, but also quite a variety of other antibiotics as well, including oxalic acid, oxytetracycline, and ninnanomycin. So which crops are people spraying the antibiotics on? Well, overwhelmingly, we found in our data that rice was the most commonly protected crop with antibiotics, as you can see here. So the point is the fact that these critically important medical antibiotics are being sprayed onto crops, and the danger is that resistance will spread from plant pathogens into human pathogens, or it would just be increasing our contamination of our world with these antibiotics. So there's a great deal of concern about this, hence it's being included in the One Health AMR portfolio. And with that, I'd just like to thank you and thank the organizers for the opportunity to talk to you today.
So our next speaker is Pankaj Dhaka from the Center for One Health, the College of Veterinary Science, through Angad Dev Veterinary and Animal Sciences University. And he's going to speak with us about addressing AMR in resource-limited regions. Pankaj, the floor is yours.
Yeah, thank you, Lisa, and good evening from India. So although the topic is very broad and multifactorial, I will try to make it as concise as possible. Please, next. So as already Dr. Craig spoke in detail about eco-evolutionary process of pathogens, and it's completely natural, but the point of concern is we are making it much faster. We are accelerating this process at a much faster pace than expected one. Next, please. Yes, this is again the famous data set systematic review of 2022 by AMR collaborator. But what I like about this data set is now we can compare it. We can communicate in more confident way that the problem is there. Now it's more visible. Previously, it was considered as invisible pandemic, but now we are having one data set to compare with other existing problem. And it's very, very important, especially to communicate to the policymakers so that they can consider it a evident problem. Next, please. Within this study, you can appreciate that the burden is huge comparatively in Southeast Asian and African continent. Next, please. So the crisis is there, and it's going to loom more and more if we are not putting any check properly. So now at present 1.3 million deaths, one child die every 3 minutes because of sepsis conditions related with resistance organism, and it has been projected that by 2050 20 million people will be under poverty, and it can cause around 1 trillion additional healthcare related cost, and it may lead to 7.5% decline. So it's affecting everyone. Next. So as Dr. Tedros rightly stated, that it's a global health emergency condition, and it's involving multifactorial component, and we need to address it. We will see. Next, please. So this is that data set on the drug resistance index. So you can appreciate data is available from European and USA, European countries and USA, and you can see very dark blue color representing that more drug DRI, drug resistance index in India. But the worrisome thing is that we don't have data from African and most of the Asian countries. So that's really worrisome. If we don't have proper surveillance mechanism, we can't communicate, or we can't bring up evidence-based solutions. And again, the problem, we must acknowledge that if resistance is anywhere, it means resistance everywhere. Next, please. Yeah, we are closely related. Resistance bug anywhere means everywhere. Next, please. Yes, it's a very, very complex process, and really difficult to communicate properly. Generally, of course, if we, in very simple terms, if we are using drug more, means more resistance, but the consumption of drug directly or indirectly depend on many factors, starting from burden of infectious disease, socioeconomic factor, political reasons, stewardship, capacity building, and many other. Next, please. Okay, this is the study by Kellin and co-workers, and they estimated the consumption between 2000 and 2015, and of course 65% increase in antibiotic consumption, but mainly driven by lower and middle income countries. But the paradox is, even they are consuming more, but again we need to consider there is unavailability of even the first line drugs among these countries. Next, please. Yeah, this again, the study is clearly highlighting the upper side. You can see that, of course, there is use of watch and reserve category of drugs as described by WHO. But the concern point is, even there is mortality without the antibiotics in most of these regions, African and Southeast Asian regions. You can see dark red in African continent. So we need to address both things, even access and unavailability, that is access to the basic antibiotics. Next, please. Of course, I won't go in detail about major contributing factor. We all know overuse, misuse, poor access to the quality medicine, diagnostic, then poor IPC, lack of hygiene and sanitations, lack of new antibiotics, of course, over agricultural practices, trade. But in right side, you can appreciate the major component, that is human, animal, and environment interface. Okay, the bugs and genes can move very fast if we are not taking proper stewardship. Next, please. Yes, of course, most often this animal husbandry often blamed for higher usage. That is around 70% of the antibiotics. Next, please. But blame is not the solution, because in coming times it going to increase, means because now the GDP in even in LMIC is also increasing. So as the purchasing capacity of the consumer is increasing, there will be more demand, and it will lead to intensive livestock production and more antimicrobial usage. Next. Now the problem is, we don't have one thing is we don't have proper surveillance system, and the second thing, we need to address this transmission links. So again, this study again estimated the resistance, and to be worrisome, that threefold of the resistance has been increased between 2000 to 2018 among all the sectors, major animal husbandry sectors, that is our poultry, piggeries, and dairy sectors. This is mainly in this lower and middle income countries. Next, please. Yes, this is often neglected. The animal husbandry and environmental interface. So Dr. Andrew already spoke in detail, but again, the concern is around 75 to 90% of the antibiotic administered to animal used to excrete unmetabolized, and these residues can pose a great concern and selection pressure. And 70 to 80% of the antibiotics given to fish again excreted to water bodies, and agricultural practice, especially discard, not proper discard of waste, including manure, again can be the source of bacteria as well as genes. Next, please. This is very recent study by Zhou and co-workers, and this again highlights, as we know, like most of the cities, especially in developing countries, there is high level of pollution. Now there is clear association between particulate matter and it's causing the selection pressure, may activate some of the genes related with antimicrobial resistance. So this study again highlighted that the things are not good, and we must put up the multi-sectorial approach to contain it. You can see it, this even the AMR derived from particulate matter is causing 0.5 million premature deaths and around 18 million years of life loss with huge socioeconomic losses. Next, please. So there is completely good analogy between AMR and climate change, and these are going to target resource limited region. Next, please. So one thing is there is no new drug, and of course new drug development is not easy task. And again, the second most important thing is if there will be development of new drugs, they are not going to reach soon to the population which need them the most. Next, please. Again, the study this clearly highlighting that there is completely correlation between governance, corruption, resistance, and infrastructure. Of course, the corruption and governance is directly or indirectly responsible for capacity building and surveillance activities. Next, please. Yes. So it's rightly termed as invisible pandemic in many sense, and there are many important parameters, especially in resource limited regions, maybe in other regions also, which are highlighting the concern. The first thing is there is limited awareness. Next, please. Yeah, limited awareness, diagnostic challenge, low lab capacities, and of course, most important, this concept is very complex to understand and communicate. It's look easy, but not easy in the field. Then there is variable resistance, and resistance rate can vary among microbes within region. And I want to highlight that we can't make one set of SOPs or package of practices which can implement everywhere, and it's really difficult to communicate. Next, please. Yes. Again, the surveillance, it's very important and lacking in most part of the world. And because if we don't have data, we can't speak, or we can't have solid evidence to motivate the policymakers. Okay, so most of in resource limited regions, there is lack of data, mainly at animal environment interface. There is lab quality issues, no link of microbiology and clinical data, bias of available data, data sharing issues, and of course the duplication of data. Next, please. So as Professor Jacob in initial lectures, he clearly mentioned that this is multifactorial. There are many factors apart from usage which need to address, and especially it depends on region to region which set of practices we need to target. But there are many factors which some of you can see, some of these factors you can appreciate on the slide. But apart from this also there are many factors, and here in India we are using participatory approach to address these issues. Linking the component of animal, human, and environment at our best, but it's sometimes behavior problem is very important, especially among the farmers, to motivate them to use the antimicrobials judiciously. So next. So thank you so much. Thank you for the opportunity. Thank you, everyone.
Thank you very much, Pankaj, for this look into the research in some resource constrained settings. And we're going to go now to the questions. We have a couple of questions. First of all, I see a hand. Deepthi Vijay, can I invite you to turn on your microphone and share your question with us? Can you hear me, Deepthi Vijay? You can go ahead and turn your microphone on and ask us the question if you have one for us. Okay, while you're doing that, then I'll move forward with a question from Luis Maya. Has anyone tried to estimate drug resistance index for countries with no actual data using econometric models and instrumental or explaining variables from countries with data? I'm not sure what the interface is telling me. One of our panelists has an answer, I think.
Of course, I'm not expert in this field, but what I can suggest, they are estimating this drug resistance index with whatever data they are extracting from the literature. And for example, in India, we don't have so robust surveillance system, but many studies are going on in compartment wise among the academics and all. So I think they are using that data and forecasting the scenario.
It's Pankaj, that's interesting. Are you aware of anything using econometric models?
No, this is not my area, sorry.
Okay. Then I think there's a related question from Emmanuel. What is the estimate cost involved in AMR worldwide? Anybody want to hazard a guess at that estimate?
I think the O'Neill report was in the trillions.
Okay. I guess it doesn't really matter whether it's 1 trillion or 10 trillion. If it's trillions, that's already a crazy number.
Yeah, it's a big number.
Thanks, Andrew. How are the other countries doing surveillance in crops, and which authorities are responsible to implement crop surveillance?
Talking off the top of my head, I don't think anyone's doing surveillance. So what we know is from isolated academic studies. So I don't think there's anything systematic at the moment.
Yeah. Okay. Yeah.
Hosts and panelists.
We had a few questions. Well, first of all, maybe a question from Jakob. I'll let you go ahead, Jakob. You had a question. Andrew, you seem like you're in the hot seat with most of the questions coming on the environmental aspect. Go ahead, Jakob.
Yeah, I hugely appreciated your environmental monitoring data. That is very important. But should it not be systemically linked to actually human and animal health and food chains? In my perception, with a few exceptions, we don't really understand the systemic distribution and spread of AMR between humans, animals, water, and the environment. Do we?
Well, we understand it from a schematic point of view, so we could draw arrows. But the weight of, I suppose what you're asking is the weight of the arrow the same everywhere? And the answer would be no. And can you put a number on it? We could if someone spent the money to actually measure it.
Yep.
And back to the last question, is that people aren't spending the money to measure it yet. But We could, but I think that maybe it's not an entirely helpful answer because you kind of want to do it anyway. And so what you probably could do is put confidence intervals on a sort of a mean estimate and just say that most places probably fall within this range, and then you could devise a model that way. But again, it's a big task to do that.
Yeah. But I argue the hypothesis is that if we understood the systemic drivers of AMR, we could really control it at the source, and that would save actually these trillions. And I wonder if the loss that we currently incur in human lives and animal lives and money, is it not worth that systemic analysis and actually identifying these major drivers?
Absolutely. You don't need to take my word for it. Someone should actually do the study to show that I was right or wrong. I agree with it.
Interestingly, I made 2 proposals to ERC and to the Swiss National Science Foundation examining exactly that, but it was always rejected.
It wasn't me.
Yeah.
Increasing complexity. We have one more question that came in with the pre-registration, so I'm not exactly sure who's the source of it, but the question was, what role might One Health play, especially on food safety with regard to food testing? And I wondered, Chris, if you might be able to give that one a start. Talking a little bit about the role One Health plays on
yeah. Well, I suppose the answer to the question is that the testing brings the whole One Health issue together, because what you're doing in the testing is you're monitoring. So in the UK, as an example, we monitor retail packs of meat, and we monitor carcasses at the abattoir, and we're looking for levels of antimicrobial resistance and what they're resistant to. And what that information provides us with is a general trend of where we're going with the whole antimicrobial resistance issue in terms of the animal's contribution. But of course, if there's a potential link then to the human population, that's kind of like a sense check as to what those risks might be. But I would say we need surveillance across the board. So Andrew was talking about the environment, and Rumour has just commissioned a report to look at what the implications of our environmental footprint are in terms of using medicines in livestock, so that we understand that. And the results or the survey seems to come back to say, well, we think there's issues there, but we need to understand much more about it, and surveillance could play a big part in that. But the debate is, what exactly is it we would need to look for or measure, or what surveillance would we do? And as Andrew just mentioned, it boils down to cost. So in summary, I think just as we found monitoring the levels of use really important in terms of guiding the decisions that we've taken, I think understanding the levels of resistance and any specific nuances within that levels of resistance would be really good. If I may, I've got a question to pose back to the panel. When we talk about resistance, is it an on-off, yes-no answer, or is it possible that you can find resistance, but that resistance isn't critical to patients or to society as a whole? So you can find resistance at low levels, but it isn't critical, but obviously we need to monitor it and make sure it doesn't become critical. Question.
An excellent question.
Is there anybody who would like to respond to that?
Oh, I can have a go. You want? Yeah. So not all AMR is created equal is I think the point you're trying to make, and that is true, and that is probably why one of the many reasons why surveillance has been slow to start is because everyone wants to look for something different, and they want to do it in a different way, and standardizing it is, the effort to try to standardize has that inherent fundamental problem of needing consensus, and It's not that we don't agree that some things are more important than others. It's about, it's also this sort of a multi-dimensional space of cost, time, resources, reagents, depending on what part of the world you're in. So, I mean, there's a lot of dimensions to it. And so do you want to create a surveillance system that is only for the UK, and therefore it's not resource limited in many other ways that it would be if it was somewhere else? Or do you want to harmonize it and have everyone use the same system, but recognizing that you're not actually looking at that question of, is AMR getting worse or better, in a sophisticated way that we could actually ask if we wanted to. So there's trade-offs, and I think that's kind of where we are in this space, is that some genes aren't as particularly useful, but they tend to gather a lot of interest because we know a lot about them mechanistically. And yeah, it's a messy space. And yeah, I have to just sort of put the hands up in that the discipline is coming to probably a consensus, but isn't there yet.
Yeah, and I think you raise the question that often comes up when we're working with integrated approaches and One Health questions. They're gnarly. You know, how do you eat an elephant one bite at a time, right? In the interest of time, I'm going to move along to our last session. Thank you very much for those questions and for the really thought-provoking answers. Our next panelist is Miss Becky McCall from UCL, and she's going to talk to us about a little bit different aspect, engaging with the public on AMR. So Becky, I turn it over to you. The floor is yours.
Hello. Hi, can you hear me? Hopefully, yes.
Yes.
Okay, great, thanks. So firstly, I would like to say thank you to CABI for inviting me to share my work today, especially on the occasion of World AMR Awareness Week. So thank you. So my work in a line, my work explores something about exploring and communicating the concepts of AMR to the general public using the medium of digital storytelling. Now, I imagine that might be a sort of alien concept to many people, but hopefully you'll find out a bit more through this short talk. And by concepts, I mean things like, oops, can we flip back slightly? Yeah, thank you. What I mean by concept, things like foregoing an antibiotic, an individual foregoing an antibiotic for the greater good, for society, preserving those people who really need them, or the idea that we're all vulnerable, you know, whether we have underlying disease or not. As far as for what I do, I spend about half my week doing my PhD at UCL, and the other half I'm a medical journalist. And I've actually been juggling these two roles for about 5 years now, and I hope to finish this PhD in about another year. So, next slide, please. So how do we currently communicate or engage with the public about AMR? I'll skim through this quickly. We're all familiar in our field. There has been a lot of attention over recent years. In the UK alone, Dame Sally Davies championed the issue back in 2014. Shortly after, there was a review, already mentioned the Jim O'Neill review. You know, some really hard-hitting figures there about what could happen if nothing's done by 2050. And then more recently, we had that Lancet report with the 1.2 million deaths caused by AMR. So lots of facts and figures. We've also had, you know, a multitude of campaigns globally and in the UK. Public health campaigns. I was looking this morning. European campaigns with hedgehogs. I don't know why hedgehogs. Can someone explain? But hedgehogs who say antibiotics don't work for colds. Okay, so yeah, it's true, fine. And we've also had, there was a recent UK one, in fact, I think it's ongoing, Antibiotic Guardian, where people are asked to pledge and actually pledge to preserve antibiotics, you know, call to action. So that's an interesting approach. And then I think earlier we had the a vet talking about Emma the vet. It was an animation where Emma the vet was looking after Dottie the dog. So, you know, all sorts of different approaches there, which are interesting. But in short, all the facts and figures are great. The messages are great. They all work together as a whole, hopefully to raise awareness. But, you know, personally, where I'm coming from with digital stories is that I would like there to be a way to make engagement more meaningful, more real and relevant to the public. Because actually most of us are not directly affected by AMR, or don't perceive ourselves to be directly affected by AMR in our lives, in our daily lives. And that is the problem. The facts and figures are out there, and that's great. A lot of scientists, researchers are interested in those, they take them on board, but general public, with everything else that's going on in our lives, does it mean anything? So what I'm trying to do with digital stories is to and these are short, just quickly, audio-visual stories told by somebody who has an experience of a drug-resistant infection, okay? And I'll go into that in a bit more in a minute. And by doing this, using the first person and the storyteller to relate their experience, I want to close what I consider a psychological distance. Okay, so this is a distance where a lot of people feel AMR happens out there somewhere to other people, probably in the future, not in my lifetime, not in my life. And to close that to the fact that, well, actually, we need to realise it could happen to any one of us. And it is affecting all of us, even though we might not perceive it that way. So what I want to do is explore how we can create these digital stories. And I would like to change the way you think, the public think, and engage with the issue of AMR and their use of antibiotics. So the next slide, please. So what is digital storytelling? Okay, well, in a line, I guess what we're trying to do is capture real life with digital storytelling. So it's capturing the lived experience and being able to relate that, the lived experience of AMR in our real everyday lives. I like to think of digital storytelling as an invitation and to invite you to dive in, to swim through someone's memories of their experience of a drug-resistant infection. And importantly, to swim through the emotions that come with that. And that is really what's very different here, is the emotions involved. So simply put, mechanistically, if you like, a digital story is a 3 to 5 minute compilation told in the first person. It ultimately aims to produce an authentic story. Okay, so it tries to get as close to the bone as possible. And by this, I mean, this is closing the psychological distance between the teller and the viewer. And if we think traditionally with films or documentaries or even animations, you've got a filmmaker, say, you've got a producer or a director that tells someone to say something a certain way or whatever, and you bring in different effects and things. This all puts distance between the teller and the viewer. So with digital storytelling, what I try and do is I actually train that storyteller, that person who had the drug-resistant infection, to create their own story, to use video editing software. And what I actually do is I take them through a full step process. As you see on the screen there, the finding, the telling, crafting, sharing. The finding is actually probably the hardest part, because people tend to tell through a sequence. I had this scan, I had this injury, I had this scan, I had this GP give an antibiotic. It didn't heal, I went back, went to hospital, I spent this long. That's just a sequence of events in one ear out the other. So what we're trying to do is actually find a moment there that is really meaningful. It's almost like that moment becomes a lens, and through that lens we bring in the emotion and we see the story of what's happened differently. So that's what then the next stage would be telling. So that is sort of writing a script, really fairly lengthy, but you'll be amazed. I've been amazed at how actually most people I've worked with can write a script really well. Okay, and it's a co-creative process. We work together. And then we add in photos, and importantly, it's all about authenticity. So we go through someone's own archive. We all have mobiles now that take photos or short clips and videos. It's a fantastic opportunity to say, go back through your photos, jog your memory, find some great illustrations of what's happened. And then we move on to the screening or the sharing, which is what I will be doing hopefully next year with a small series of stories. Because essentially it's about real people, okay? It's about real stories and real experience. And it takes about 8 hours, I suppose, of one-to-one work. Okay, next slide, please. Right, I love this because this just really sums up what digital storytelling is all about. Okay, so this is a quote by a Hollywood scriptwriter called Robert McKee. He's well known in the storytelling world, and he says that really the powerhouse of creativity mean, whether it be digital storytelling or writing a script for Hollywood, really is about connecting to seemingly unrelated things to create a third thing, a thing that is unknown, that is new. And in terms of digital stories about AMR, what we try to do is we create the health part of the story, the drug-resistant infection part of the story, and we connect that up with something else in their lives. And when you look at it, you might think, well, that's totally unrelated. But it might be something to do with a family, or maybe their career, or a place they like to go to, a hobby. But we connect these, and then you come up with your digital story, and you interweave the two. And that second thing will help illustrate the emotion. You know, we're not all Charles Dickens, we're not all great script writers or novel writers, so we find other ways to illustrate those emotions. And one way to do that is through photographs, film, dialogue, excerpts from plays or music, all that type of thing. And you add that into your 3 to 5 minute story. And I also think that this crossover between two things, the health and the other part of your life, is really, really important, because as I said before, not many of us have direct experience of a drug-resistant infection, of AMR. So what we try to do is we build a bridge. And we build a bridge between that AMR experience and another part of their life, their hobbies, say, or place they like. And by doing that, perhaps a viewer can relate to that other part of their life. It might be their hiking, or their love of dance, or music, or whatever it might be. And that way you draw them in and you relate to your viewer. And that hobby or whatever it might be will illustrate a feeling that goes with the experience of that resistant infection. So it's building bridges and it's connecting. So I do have a story, actually, that I'm really, really pleased to say that the first story was completed yesterday, and we actually released it yesterday, and it is viewable on the Medical Research Foundation site as well. So we might get a chance to look at that if we have time. So yes, next slide, please. So just in summary, I am completing a series of 5 stories for this piece of PhD work. And then I will hold a screening and a discussion following that with about 8 to 10 people, I think, and that will provide primary data for analysis. And the prime question is really going to be, you know, it's going to be what value does DST, digital storytelling, bring to understanding and insights on AMR by the general public. So the next step is going to be, beyond that screening, is how we use digital stories as part of a larger public health communication effort. So right now, that is an open question for me, and I really welcome any comments. I personally see digital stories could stand alone. Someone said to me the other day, you know, they could actually just be a little sort of installation-like art piece on their own, some of them. But I like to see it as something pragmatic, something we can use alongside other communication methods. Okay, so I would see an integrated digital communication campaign, and really working alongside. So the value that therein lies the value, really. I also think, finally, the material in these digital stories offers some new insights, it's in depth, and I wonder if that is raw material that could actually feed a campaign. We see posters, radio ads, TV ads, social media ads, public health communication ads, whatever. They all need text, they need images, they need clips, whatever it might be, it's all there. And that's really, that's what I have to say. And as far as, just quickly on the One Health side, I wanted to explain the process, and there is no reason why this process cannot be used in the veterinary world, the environmental world. We could talk to farmers, what are their stories? We could talk to vets, what are their stories? You know, there's scope here. So I don't, do we have time to watch a bit of the film, please?
Actually, Becky, thank you very much for showing us another side of the awareness campaign. We're out of time for right now. I'm going to go ahead to the last speaker, but when we get back to the questions section, if we are finding extra time, then that would be a great opportunity for you to show us a little bit about the video. In the meantime, maybe you can share the link so that the participants can see that link. And with that, I'm going to go ahead and invite our last speaker, Dr. Boma Ezekiel Prosper Noali, who's working with Youth Actions Against AMR and as well the WHO Quadripartite Working Group on Youth Engagement for Antimicrobial Resistance. And so I'd like to invite you to go ahead. Boma, the floor is yours.
Hey everyone, I don't know if you see me very well. Do you get me?
Yes.
Okay. Thank you very much for this opportunity to present our actions with Ajiram, with Action Against AMR. So that my presentation will be focused on best points. I will talk to you about Ajiram, our current work, our challenge, lesson learned, and level to activate to engage more young people in the fight against AMR, and also my call to action. Slide, please. So about Ajiram. Ajiram is a multidisciplinary organization of young African leaders in Africa. We set up this organization in 2021, and our network is present in 8 countries now. We have Burkina Faso, Benin, Guinea, Togo, Mali, Niger, Senegal, and Chad. And our vision is to ensuring safe and effective antimicrobial for future generations. Slide, please. So I will talk about our current work. We have some activities that we realize to engage more young people in the fight against AMR in Africa. First of all, we have educational talk in school because we know that education is the most powerful weapon with whom we can use to change the world. So that we exchange with young people in schools on AMR, on 1F and infection, and our methodology consists to present some videos, and we follow with exchange, and the most participant young people during the session will get some gadgets. Impact of these activities that for this year we reached 1,000 students with this activity. Next slide, please. We set up 1F close in school because we know that 1F approach work because it addresses health threats from all possible angles, so that we set up some clubs in school that permit to young people to be more trained about some topics related to AMR and also to 1F. And our methodology consists that the members of the school prepare something to present a topic, and so we invite some specialists that came and exchange more with the members of the school during the session. We set up 4 clubs in Burkina Faso now. Okay, we have also AMR Youth Leadership Program, because if you are actually inspire others to dream more, learn more, do more, and become more, you are a leader. So if you want some change, some impact in the fight against AMR, it is very important to raise a generation of young people that are leaders, that have the capacity to influence, the capacity to change the world. So that this program consists to empower young people during 4 months. And for this year, we received 600 applications, and we select 200 young people from 15 countries. And we continue in the training of these people. And also we have a program of mentorship after this program. Slide, please. And also, also we have some activities that consist on sense activity conference, because sense is a key to combat AMR. So that we organize some conference and we participate from to some workshop with some stakeholders to give our point of view about how young people can be more engaged in the fight against AMR, and also what is the area that where we can work more. Slide, please. Slide, please. Okay. Also, we communicate very much in AMR. And also we have some strategic partnership. You see in this slide, we have a strategic partnership with French Embassy. We have strategic partnership with some other organizations like Rambukina, like RIAC Africa. We will work together in the fight against AMR. And also we are part of the quadripartite working group on AMR. Slide, please. So with this quadripartite working group on youth engagement for AMR, I am a member of this working group, and this working group consists that to engage more people in the fight against AMR. We have 3 objectives that we perceive in this working group, that to increase the visibility of young engagement in global AMR action, and also to strategize and invest in activities which can raise the awareness about AMR, and we also to activate, reach, and connect with other young people around the world to amplify the message around AMR, and also advance the advocacy priority on the working group. Slide, please. So what is the challenge in this commitment? You know that AMR is little no problem among our population, and even among health professional and government officials, specifically in our countries in Africa. And also, AMR is not a priority of some countries' planning. This is a serious problem. And young organizations are not very involved in AMR in sub-Saharan Africa. And we have also the resource limitation like time, money, human resources, etc. that are the challenge that we have in this fight against AMR in our countries. Slide, please. So that, what is the lesson that we learn and the levers to activate? AMR concern everyone, and we must apply the One Health approach in the fight against AMR, so that we must involve civil society, journalists, community leaders. The second lesson we learned, that without strategy of action, no lasting results. Youth people need to work together. And so that I think that we must set up a collaboration network on youth AMR in Africa and around the world, like the quadripartite working group, organize some annual event for youth, and also strengthen youth people leadership skills. Because if you want that the future generation be a generation that will be more engaged in the fight against AMR, you must strengthen the skills of this generation. And also, we have a lack of resources to limit the progress. So that we must invest in innovative youth projects. This is very important. Also identify and provide opportunity for ambitious and community youth people, like scholarship or funding. Slide. And also, we think that education is the most powerful weapon to change the world. So that we must invest on educational tools in school, and also advocate to include AMR in the teaching materials in school and university for all disciplines. And also, we know that no communication, no action, no change. It is very important to amplify the communication about AMR, and also to develop some strategy that can permit us to optimize antimicrobial use in human health, animal health, and all the areas that are concerned by AMR. Slide, please. So my call to action is that everyone must do his part. You know, this woman, it is a man that inspired me because Yakuba Sawadogo is a woman that I call the man that who stopped the desert. He fights against the desert in silence in his village without funding, but his conviction made him an alternative Nobel Prize winner. So that this is a conviction, and we must do our part. And the duty to act against AMR call us like a soldier. Let us respond properly and let us act. Be a AMR champion. Thank you very much, and I'm so sorry for my poor English. Thank you.
Merci, Ezekiel. On est très reconnaissant pour ta contribution. Thank you very much, Ezekiel. That was a great contribution. My question was, is there any way that you can measure the effect of your education? Can you, for example, measure increase in knowledge, attitude, and practices or something like that? Je te traduis. Est-ce que tu peux savoir si votre engagement a vraiment un effet? We don't hear you. We don't hear you. I'm sorry, we don't hear you.
Can you unmute your
please,
microphone?
Please unmute. Yeah.
Do you hear me?
Yes.
Now we hear you.
It is good now?
Yes. Go ahead.
Okay. So that before to organize some session, educational session, we have a
Okay, we wait for Ezekiel to come back. I think there was another question to Becky.
Okay.
Can we just check? Yeah, go ahead. Lisa.
So the question actually comes from Jakob.
So that we see how the knowledge are more ameliorate about before after the training, so that we have some data on this session that we organize, and also with some laboratories in Burkina Faso. We have a reference laboratories of AMR. We are working to set up some studies on knowledge, attitude, and practice of students on AMR, and also on the young professionals on AMR. And we are working on it, and we have some results that we can share to you after if you need it.
Thank you very much, Ezekiel. Please, Lisa.
So one last question, and then we're going to say goodbye from Luis Maya. He says that these are very interesting strategies to raise awareness, both storytelling and youth leadership. And he wonders how much we can help people better understand that AMR involves decisions under risks, which is something that we're generally not cognitively prepared to do. Maybe, Becky, I can address that question to you. What answer?
Yeah. Okay.
Yeah.
I think there's a lot to do with perception of risk. Okay, and that's very different to each of us. I think. Okay, so one of the big problems is we don't perceive that we are at risk. We don't perceive a threat from AMR. Okay, that's generally it's to the point that I made earlier, which is, you know, if it's not directly in our lives, you know, a post on the tube that says, oh yeah, you know, antibiotics don't work for colds. Yeah, well, fine, yeah. And then we read something else. You know, it doesn't really drive the message home. And I think that part of that is the fact that we just don't see there's a risk or a threat from AMR. And I think actually if with storytelling, just speaking to that, I think that we can illustrate through the emotion, through the ways of connecting that I described, hopefully we can make it more real for people. And yeah, it might not help them. They might not come across a drug-resistant infection in directly themselves or someone in their family, but we are all at risk from that because of the nature of AMR, which we all know. And I think I would hope storytelling would go some way to making it more real for people, because naturally as humans we relate to the emotion and we relate to stories. We always have. It's ingrained. It's part of who we are. We grew up with storytelling.
Yeah, so we have to address these questions on different levels with different modes of communication. Very, very true. All right, in the interest of time, I'm going to turn it back to Jakob, and I just wanted to say thank you to all of you for joining today, and I hope that you all have learned something. I certainly have, and I appreciate all of our panelists who contributed today. Jakob.
Thank you very much also from my side to all panelists, but thank you also very much to the team of CABI, in particularly to Georgina, who have set this all up. And we look very much forward to the next seminar. I think we will going to offer you a series of One Health webinars of many different topics, but surely we will come back to antimicrobial resistance. I mean, my big take-home message is that I have seen that antibiotic resistance kills now more than it kills per year of malaria or of TB. And that's really impressive for me. So let us continue. I think One Health has a lot to contribute to improve that situation. So we look forward to interact with all of you again. Thank you very much and all the best to all of you, wherever you are in the world. Goodbye.